finish our lecture on the cell, or at least let's get a little bit farther through it than we've gotten so far. Zoom in here. I'll move my computer screen over a little bit more. Oh, there we go. Okay, you guys, well, let's just pick up right where we were. We just finished this, the plasma membrane components, and let's keep going. Except now I can't find my other one, of course. Okay, oops. Okay, so membrane transport. This is kind of the big one that we're going to get into a little bit more detail than what your book has in it. So, there's basically two types of transport. When I say membrane transport, we're talking about the plasma membrane. Okay, so the plasma membrane has a really, really important job in the cell. And like we said, it's selectively permeable, so it lets things in and out the way that it needs to. And so, in some ways, we're going to spend a lot more time. We're going to spend a lot more time on the plasma membrane than we will on a lot of other parts of the cell, and that's just because anatomy-wise, like when we're talking about the function of the body and how it works, a lot of that depends on the cell's ability to take in what it needs and get rid of what it needs. So, okay, so there's two. We can kind of divide this out. We have two types of passive transport. So we have, we have two types of transport. I'm sorry, we have passive and active, and you guys may have heard of these terms before. Passive, it means that there's no energy required. Things can just move in and out of the cell as they need to. So two types of passive transport are diffusion and then filtration. Active transport does require energy. It requires ATP. And if you guys will remember, ATP is just it's the energy that our cells use to move things, for everything. It's just like packets of energy that our cells use to perform functions. Okay, so there's basically two types of active transport in the plasma membrane, and that's solute pumping and bulk transport. And we'll talk about these in detail. And actually, one thing that I think would be really helpful for you guys is if you guys will grab one of the old textbooks just to look at for notes today. If you will do that, go ahead and grab that, and you guys can turn in there to, it's like page 67 and 68 and 70. That's kind of where we'll be. We'll start on 67 and talk about diffusion. Okay. So grab those books, and you can push pause for a second until everybody gets there. And let's talk about a few vocabulary terms first. Okay, so first is a solution. We talked about some of these in the chemistry chapter, but a solution is just a mixture of two or more things. Okay, that's it. So you put salt in water and you've got a solution. A solvent is the substance that's present in the largest amount in a solution. So if we're making salt water, let's say I have a cup or I have a glass of water and I'm pouring salt, the water would be my solvent. Okay, it's the one that's in the largest amount. Okay. So in the body, water is our main solvent. Remember we talked about our body's like 60% water. So that's our main solvent in our body. Solutes are the substances present in larger amounts. So if I'm making a glass of salt water, water's my solvent. The salt that I'm putting in is my solute. Okay, then we have intracellular fluid, like we talked about, ICF. Okay, this is in the cell, so it includes the nucleoplasm and then the cytosol. Nucleoplasm is like the plasma, or the kind of like the fluid within the nucleus, and then cytosol is the fluid that's inside, like the cytoplasm. Remember we talked about the three main parts of the cell are your plasma membrane, and your cytosol, and then the nucleus in the middle. So, your cytoplasm. So your cytosol is just like the fluid inside the cell. Okay. So look, it contains small amounts of gases, that should be nutrients, and then salts. Now, the interstitial, if you'll notice, remember intra is inside, inter is between. So interstitial fluid, means between the cells or between tissues a lot of times. This contains nutrients, hormones, neurotransmitters, salts, waste, anything that's been moved out of our cell, okay, and then, or that needs to move in. So the cells can kind of exchange. Interstitial fluid is, it really means between tissues, not cells. But anyways, that is a specific type of ECF or extracellular fluid, like we talked about, okay? So the cell's able, all the cells kind of flow freely in this extracellular fluid, and this extracellular fluid's got extra of everything that it needs, that the cell could draw off of or get rid of. Okay, so again, it's selectively permeable. Let me move my little picture here so you can see this picture. Okay, so just like the picture we looked at in our textbooks, allows some things to pass through, some things it doesn't. This only happens, like I said the other day, in unharmed cells. Remember we talked about like burns or any kind of damage that is done to cells. It makes them more permeable, and so things are allowed to move through that really shouldn't move through. So movement through the plasma membrane occurs passively and actively, like we just talked about. Sorry, kind of a repeat slide here. Okay, so first one is passive transport. Okay, passive transport. Diffusion is like your most basic transport. Okay, this is, like it says, basically every cell in the body uses this at some level. Okay, molecules scatter in available space. So for example, let's say you've got a glass of water and you put a drop of food coloring in there. Okay, that drop of food coloring is really concentrated. That's why it comes in little bitty tubes. Okay, but if you'll notice, once you put it in the water, it like disperses. And that's because that water had no solutes, none of that food coloring in it before. So that food coloring that's really concentrated then spreads out so that it diffuses so that it can spread out and not be so concentrated. So eventually, all the water would be, theoretically would have the same concentration because everything's going to move to and from.
Tasia Brown, if you're in the building, please come to the office. Your grandmother is here to pick you up. Tasia Brown, come to the office.
Okay, notice it says it moves down a concentration gradient. So your concentration is basically like your strength of the solute in the solvent. So it'd be like, if we're talking about my jar of salt water, my glass of salt water, it'd be your concentration is like the strength of the salt in the water. Now, so speed is controlled. Anyways, let me finish that. Down a concentration gradient. So what it's saying is things are automatically, if we have a permeable membrane, okay, things are automatically going to move from an area of high concentration to an area of low concentration until they're equal. So another example would be like Alka-Seltzer or something. So you put a little thing of Alka-Seltzer in your drink and it disperses and it diffuses because it's really concentrated in that tablet, but the water is not really concentrated. Okay. Says speed is controlled by size and temperature. So what do you guys think happens if it speeds up? Never mind that question. If it heats up, okay, it's going to speed up, same as metabolism. Like plasma membrane, that PM stands for plasma membrane, by the way, says it allows passive diffusion if the molecules are small enough or if they can dissolve in its fatty portion or that phospholipid bilayer. Okay, so passive diffusion can occur if the molecules are small enough to fit into the cell between that phospholipid bilayer, or if they're soluble in that fat. If they can like be dissolved in fat, then they can easily move through that fatty layer around the cells, the phospholipids. Okay, so that means things that are more concentrated outside the cell, if they're small enough and if they can diffuse through there, they will diffuse into the cell. Basically with diffusion, what wants to happen is for both sides of that to have the same concentration eventually. Okay. So look, it says glucose and oxygen continually move into cells because the cells use them up. So you've got this membrane, the outside of the cell, and they're constantly pulling in oxygen and glucose because the cells, in order to make energy, is using up the oxygen and the glucose. So there's constantly less concentration of glucose and oxygen inside the cell than there is outside the cell because it's constantly using it. So there's no more available. Anyways, I hope that makes sense. If not, we'll talk about it. Write down your questions and we'll talk about it whenever I get back Thursday. And then CO2 is just the opposite of that. So our cells, after they use the energy or create the energy, okay, using the glucose, then carbon dioxide is a waste product of that. So there's always more carbon dioxide in the cell than there is out. So it's constantly being pushed out of the cell, not pushed. It's just constantly moving out of the cell by diffusion because, there's more of it inside than outside because it's continually being made. Okay, so there's two types of diffusion, actually. We have simple diffusion and facilitated. Simple diffusion is just things move through that phospholipid bilayer, the plasma membrane around the cell, very easily. They just move through. So anything that's fat soluble or that can dissolve in fats, because remember that phospholipid are fats. So anything that's dissolvable through there, anything that can fit through really tiny pores in the cell or through the cell. So it says chloride is an example of that. Oxygen and carbon dioxide are both examples of that. Okay, another example of simple diffusion is osmosis. Okay, and osmosis specifically refers to water. Okay, so water specifically refers to water, osmosis does. So water will move through this membrane around the cell, but it moves through a protein channel. Remember we talked about aquaporins, can't talk. Those are these little water channels, so things can move through, but that's osmosis. It doesn't need energy, it just moves through. So if there's more water outside the cell, it's going to move into the inside of the cell. Osmosis, when you hear osmosis, think movement of water. Okay? Osmosis, movement of water. Osmosis, movement of water. Okay? Anyways, I always want you to think to pair osmosis with water. Okay, one other thing to think about, and we'll talk about osmosis later this week too, but I don't have a paper with me. But water always follows solutes because it's trying to make everything equal. So for example, let's say you have a day where, well, this is an example, salt makes you thirsty. And that's because water tends to follow the solute. So the more salt you take in, the more water your body's going to require because that salt kind of pulls that water towards it. So because of that, you're left with a low water supply because it's all kind of attached, if you will, to that salt, if that makes sense. Anyways, kind of crazy. But notice too, it says that osmosis is diffusion of water, but it moves through a protein channel because of its polarity. So it may have a harder time moving in because of the way that it's polarized. Okay. Try to see if that talked about osmosis in this book, in your new book, but I don't see it. We are actually going to talk about in detail about osmosis next week. We're going to do a whole lab over osmosis. Really cool, fun lab. I think you'll find it really interesting. The place where this really becomes important, our cell's ability to maintain fluids, is like when we start giving people IV fluids because they're interested in nursing or being a doctor or anything like that. IV fluids change the levels of our electrolytes and can change the levels of, change our cells' ability to soak up fluids or get rid of them and so forth. So it's really important that we understand how these things, how osmosis works, how water can move in and out, so that we can give people the right IV fluid. So we're going to look at that. It's pretty cool. Okay, facilitated diffusion. It says use protein carriers to transport. So the kind that we talk about with the channel proteins that actually carry substances across, that's called facilitated diffusion. Still doesn't necessarily use any energy, okay, but it does require an extra something to move through. So some proteins in the plasma membrane carry glucose for cell use. Otherwise, glucose can be too large. So glucose sometimes can fit through, sometimes it doesn't. I think I just said the opposite of my thing. No, sorry. Anyways, so that's an example of one that's moved through. It doesn't take energy, okay? Diffusion, any kind of passive transport does not take energy, okay? Now, this last type of passive transport we're going to talk about, and remember, we're talking about things transporting into and out of the cell, is called filtration, okay? Now, filtration is not very selective. The only place, I know this is used in a couple areas, the main place in our body where this is used is in our kidneys. Okay, so it says the process in which water and solutes are forced through a membrane by fluid or by hydrostatic pressure. Hydrostatic pressure is just the pressure within our blood vessels. Okay, so basically what happens in our kidneys is our blood vessels get smaller and smaller and smaller until they get into this little, you can kind of see this little renal, it's actually part of a nephron that's on the screen on the top right hand side, but we have what's called the glomerulus, and it's this little bitty ball of capillaries. Capillaries are very, very, very tiny blood vessels that are like one cell thick. I mean, they're very tiny. Sometimes like one blood cell can fit through at a time. I mean, they're so tiny. And inside this little ball of these capillaries, the pressure is very, very, very high. And because of that, because that pressure is so high and there's so much fluid in there, it actually pushes almost everything out of these blood vessels. So basically, everything except for your red blood cells actually gets pushed out of there, and then it's just red blood cells. Now, it gets reabsorbed later in the kidney, and we'll get to that when we get to the urinary system. But that's filtration. Things get pushed out because of pressure. Think of it like, you know those black garden hoses that people get that if you've ever turned one on, if you just barely turn it on, then water just kind of like trickles out. But if you turn on like your garden, like your faucet full blast and then hook that hose up to it, it just starts like spraying everywhere. Okay, that's kind of like filtration. It's the same idea there. There's enough pressure that it just pushes everything out. Now again, filtration is not selective. Okay, so just basically everything leaves the blood vessel at that point. So hydrostatic pressure is exerted by blood, and then the pressure gradient utilized pushes filtrate from high area to low area. Same thing, goes down the concentration gradient. And anything that's works by passive transport goes down the concentration gradient, meaning it goes from an area of high concentration to an area of low concentration. And this is what allows the kidneys to do their job because everything's filtered out. And again, it's not as selective. Okay, active transport is when energy, or ATP, is actually used to move things across the plasma membrane. Okay, so this can be used either when substances are too large to move across and there's not a protein channel for it, if they can't be dissolved by fat, or if they need to move against their concentration gradient. Okay, so what that means is that normally, like I said, everything wants to move to an area of high concentration to an area of low concentration. But with active transport, one of the things it can do is it can move substances from an area of low concentration to an area of high concentration. And it's important that our cells are able to do that because our nerve impulses depend on it and so forth. So this is actually, there's a really good picture of this on 68 in the old book. That's when he had out, actually gives two pictures of simple diffusion and facilitated diffusion. Those are really good to watch, really easy to see. Okay, and then look over on page 70. Okay, this gives you, this is an example of a solute pump. Okay, so this one in particular is a sodium-potassium pump. And these have to actually move against their concentration gradient. That's why these work the way that they do. Okay, so look at this on page 70. Let's look at this. So it gives this example of the sodium-potassium pump. Look at it. It says, number one, the binding of sodium to the pump protein stimulates phosphorylation by ATP, which causes pump protein to change its shape. So basically phosphorus, which is another electrolyte, okay, this is you don't have to know about the phosphorus as far as testing purposes go, but it attaches to this solute pump, and what it does is it changes the shape of this protein and basically makes it so that sodium will get absorbed from inside the cell, go into this protein channel, and then it will be released. As the shape change expels sodium to the outside, and then extracellular, okay, outside the cell, potassium binds and then causes the release of the phosphate group. So basically, you've got sodium, the phosphate attaches, opens up, sodium enters, it opens up to the outside, sodium leaves, potassium comes in, and then so it switches. That's why it's called the sodium-potassium pump. Everything switches, but it's switching against its concentration gradient. So it's just not going with the flow, basically. It's going against the flow, if you'll think of it that way. Now it says loss of phosphate restores the original conformation for the pump protein, and then potassium is released, and it all just happens again. Okay, so this picture on page 70 is a really good picture of how that works. But it requires the use of energy because it's going against something. It's kind of like, okay, let's say you're in think of it like this. You're in the lazy river, okay, at Hurricane Harbor. Okay, if you're in your inner tube and you're going down the lazy river and you're going with the stream, okay, you don't have to use any energy, okay? But if you turn around and start trying to swim upstream in the lazy river, okay, you're going to end up using a ton of energy. It's the same idea with this, okay? Same idea. Okay, the other type of, I should have put it over to here. Oh yeah, this solute pumping is extremely selective. It only works for certain ions, certain electrolytes, okay? So there's different ones for different things. Okay, bulk transport, there's actually two types of bulk transport. We have exocytosis and endocytosis. And again, there's really good pictures of these on page 71 in the old book. If you want to get it, just look at the picture. Okay, exo means outside, so cyto means cell. So this is the process of moving something outside the cell. And basically what happens here is cells, you have these little vesicles that's actually put out by the Golgi apparatus in your cell. And they can be, there's lots of different things that they can carry. You know, it can either cause a new, you know, it can be actually just something to make new phospholipid, to add to the phospholipid bilayer. It can excrete something from the cell. But it actually, if you can see on this picture on 71, it actually like starts out as a little bubble, a little vesicle, and it moves to the outside of the cell, and then it just kind of binds with the cell and kind of spits everything out. Okay, so that's one example. No, and this again uses energy. So endocytosis is just opposite. It's in the cell, if you want to think of it that way. The process of bringing something into the cell. So it actually does just the opposite. You've got the plasma membrane. It kind of like engulfs whatever you need, and it just kind of pinches off and makes its own little bubble into the cell and moves things in wherever they need to go. Okay, as it takes substances from outside the cell into the vesicles that then close off into the cytoplasm of the cell. And then depending on what it is, it can be fused with the lysosome and digested. Lysosome kind of works like a garbage truck in your cell. It's kind of how it functions. It says if the engulfed substances are really large, like dead cells or bacteria, then endocytosis is called phagocytosis. And there's certain white blood cells that do that. You guys may have seen that whenever you did the diversity page. Bulk phase endocytosis, this is like your typical activity. It just takes in extracellular fluids that have proteins or fats or hormones or something, and then it just pulls them into the cell so that it can be used. That also uses energy. So really kind of interesting, I don't know, kind of cool how that works. 71, I think the pictures are really good. There's something on there you don't understand, you can check out one of these books and read it, okay? Or ask me whenever I get back. Okay, good deal. All right, let's get through. I want to get through at least to the cell part. We won't get to protein synthesis quite yet. So, okay. Cytoskeleton. The cytoskeleton actually strengthens and it gives the cell its shape. So there's all kinds of different fiber types. And these are all pretty much going to be your proteins, okay, because they give something structure. So cytoskeleton, cyto means cell, so it's basically cell skeleton. It's what gives each cell its shape and allows it to move like it needs to. So 3 basic fiber types. Microfilaments are the thinnest, and these are really, they're found in most cells, but really prominent in muscle cells. The intermediate filaments, an example of this is keratin, and some of you guys have probably heard that word before, but this helps to resist tearing. So it actually keeps the skin from like tearing apart or pulling apart. And it, one example of this would be in the skin. And then we have microtubules, and these are the largest in diameter. These actually help to separate the cells and pull chromosomes apart. You guys have probably seen those when you look at a picture of like mitosis, like in your regular textbooks right now, not the old ones. There's a picture of a centriole is an example of a microtubule. There's a picture on, I don't know, 62 in your new books. So we kind of see examples of that. But anyway, kind of interesting. Okay, so organelles in the cytoskeleton, microvilli is one example of something. These really increase surface area. One place where your body has these is in your digestive system. So you have a lot of microvilli. All the cells have, actually your cells in your small intestine have villi, and then those cells have microvilli. So they increase surface area to take in nutrients or excrete nutrients, whatever. Then the cilia, okay, these are actually more hair-like. So they're typically longer than your microvilli. Some cells that have cilia are like in your trachea, or like basically all through your respiratory system, and they actually beat all the time. They, like these cilia, just beat. And what they do is they move up mucus, like into your throat, so you can actually spit it out or swallow it. They're constantly moving up any kind of fluid that gets down into your trachea and towards your lungs. And then centrioles, oh, I just said, well, microtubules is kind of, what did I do? I don't know, I get crazy sometimes. Well, I was thinking centrioles are made of microtubules, but maybe I just completely got them. Maybe I just completely went crazy. not crazy. See, y'all, I'm not crazy. Most cells have a pair of centrioles. Okay, an area of the cell near the nucleus. It says each centriole is a short cylinder made of 9 triplets of parallel microtubules. See, I'm not totally crazy. I know that. Anyways, these help in cell division. Okay. All right, it says review and assessment. So match these words below. I'll let you guys pause for a second, let everybody answer, and then you can restart the video. Okay, so 1, no energy required. Okay, that would be passive. Okay, passive transport means no energy, no ATPs required or anything like that. Okay, the control center of the cell, hopefully everybody knows this, but it's the nucleus. Strengthens and shapes the cell, that's the cytoskeleton. And then moves mucus, that's the cilia, like we talked about in your trachea. Beats. Kind of grosses me out that it constantly beats, but anyway. Oh, I'm sorry, forgot to put that slide up. Okay, let's keep going. So mitochondria, okay, this is our powerhouse of our cell. If you think of it, the mighty mitochondria, it's kind of the powerhouse of the cell. And it actually makes ATP. So it uses glucose and creates the ATP, and that's what, allows our cells to use that for energy. So my question for you is, what cells do you think would have the most mitochondria? Typically, it's muscles, okay? That's what's going to use the most energy. So energy transformation actually takes place in like 2 basic cycles. First, we have glycolysis, and this actually starts the ATP production. And this is the breakdown of the glucose. This happens in the cytoplasm of the glucose, so kind of just the gel that's in between the membranes. But basically what happens in glycolysis, glyco, remember, is sugar, and anytime you see that word lysis, typically means like breakdown or destruction. So it's like breakdown of sugar, breakdown of glucose. And that's what glycolysis is. It actually breaks your glucose into 2 different molecules called pyruvate molecules. So they're like 2 half glucose molecules, if you will. Okay. Now, then what happens is these actually move into kind of the mitochondria part, the heavier part of the middle, and enzymes actually perform what's called the Krebs cycle, which I know you guys learned about in biology. We're not going to get into depth in that, but just know that that's what actually makes energy in the cell, okay, is that movement of that breakdown of those 2 sugar, or those sugar molecule into 2, and then it goes through that Krebs cycle, and in that ATP is made. You can kind of see on this picture, you've got your membranes, and then you have these little kind of bumps looking things on the mitochondria, and those are the synthase. Remember, ACE means it's an enzyme. So ATP synthase would be an enzyme that actually makes ATP. Okay. Okay, the Golgi apparatus, this actually just makes vesicles, and vesicles, or vesicles, however you want to say that, are kind of like little storage bubbles within the cell, the cytoplasm, and they move materials out of the cell, and then they can actually make more membrane, kind of like I talked about with the exocytosis. So that can actually just make more membrane and be fused. You have they can carry proteins, they can carry, they can carry anything. They can carry hormones if they needed to, whatever they need to carry, they can. And then lysosomes, Golgi apparatus also makes lysosomes, which are your digestive, kind of like the garbage truck, like I said, of the cell. They actually kind of digest all of the bad stuff that's entered into the cell, the things that we don't need. Okay, ribosomes. Ribosomes are really, really important. I think, it's not something you hear a lot about, but ribosomes is basically the place, or that's where new proteins are made. So that's where protein synthesis takes place. When I talk about protein synthesis just means the creation of or production of. So the production of proteins actually takes place at the ribosomes. That's why it says make polypeptides. Polypeptides are little bitty short chains of amino acids. Okay, so it's kind of like a little bitty miniature protein, if you want to think of it that way. Let's look, I want you to look at real quick at the picture. Where are we? Here we are, I'm sorry. On 57 in your new book at the bottom, you can kind of see the rough endoplasmic reticulum. Kind of, it's like this little cavernous looking flat membranes, and they anyways, so they wrap around the nucleus of the cell, but on them, okay, they have, well, one part, there's two parts. We have the rough endoplasmic reticulum, or the ER, and the smooth ER. The rough one is rough because it's actually studded with ribosomes. So that is where protein synthesis takes place. It's where proteins are made in our cells. Okay. Now, make sure. And then the cisternae are flattened protein synthesis. Now the smooth endoplasmic reticulum is kind of more tubular. It's not quite so flat. And so it actually can replace membrane parts, kind of like the Golgi apparatus, sort of. It creates mitochondria. It makes the nuclear envelope, endoplasmic reticulum. So it's kind of a replacement factory, if you will, for the cell. Kind of works that way. So really, really interesting. Okay, the nucleus, pretty straightforward. It's covered by what's called the nuclear envelope. It's kind of like its own little membrane, if you will, its own little plasma membrane. The nucleus has one. If you guys still have that old book out, I think there is a picture of it on 60 in the old book. You can kind of see, it kind of gives you an example. Not really a great picture, but anyway. Okay, your nucleus contains DNA, which is on the inside of the nucleus. Really important. Remember, your DNA tells your body how to do everything and how to make proteins. And then the nucleolus actually makes what's called ribosomal RNA, which we're going to talk about in a few days, which is really important for the production of the proteins themselves. So that is that it? Did I skip something? No. We'll get to DNA and RNA. We'll talk about it probably on Thursday or Friday, and the same thing with DNA. We'll talk about that too. And mitosis. We're just going to touch on these really quickly because I know you guys have studied these before. I know that it's been a little while. So anyways, I guess if you guys have any questions for me, just make sure and write them down and let me know. And we will finish then.